Welcome back. We're going to talk today about DNA replication in the cell cycle. We'll begin with a very brief overview of the DNA structure. This should be review for you, but let's take a look at what we see with DNA. First thing you need to know that it's double-stranded. What this means in the most simple terms is that we actually have two polymers. Recall that a polymer is formed of multiple building blocks that are bound together. These polymers, in the case of DNA, are called nucleic acid. If we take a look at the figure on the right, we can see these two different polymers. And let me highlight, let's see here. We're going to highlight what we call the sugar phosphate backbone. In simple diagrams of DNA, this is usually represented as a ribbon. I'm going to highlight the ribbon, the DNA phosphate backbone of one of the two polymers. So the pink strand that I'm highlighting is the phosphate sugar backbone for polymer number 1, or nucleic acid number 1. The second DNA nucleic acid is, I'm going to highlight in green, and you can see again the sugar phosphate backbone here that I am highlighting. So you can see now that I have two polymers, polymer 1, polymer 2. Both of those are nucleic acids, and we call DNA double-stranded. Sometimes we write DNA with this DS DNA, and that stands for double-stranded DNA. There are occasionally times when DNA is a single strand where these two double strands will separate, and we'll have an opportunity to see portions of the DNA strand separate during DNA replication. It'll also do this during transcription and for the synthesis of RNA. We'll not get into the difference between DNA and RNA too much right now. I will revisit that when we talk about protein synthesis. But for reference, DNA is typically double-stranded. That's its most stable form. RNA is often single-stranded, and so we'll look at that. But they're both nucleic acids. This double-stranded DNA contains in the center these, we call them nitrogenous bases, and I'm going to show you these in more detail in just a moment on the next slide. But the bases that I'm highlighting here in yellow are going to form hydrogen bonds that will keep these two polymers, these two nucleic acids, linked together. If you move from the far right into the image that's in the left, on the left here, we see a more simplified diagram where we can see that we've just straightened everything out so we can actually look at what we're dealing with here. So again, this backbone is formed from sugar and phosphate. Okay, so these are going to be linking sugar molecules and phosphates together. Same thing with this backbone here. And then in the center we actually see the nitrogenous bases that are present in the center between those sugar phosphate backbones. These bases have different names depending on their structure. Guanine is shown in green. It's just a simple green box. This is a simplified diagram. We have adenine in red, so I'll go ahead and say, hey, it's going to be pink today. And we've got thymine in blue. I'm going to need another highlighter, so let's give myself a blue highlighter. And so my thymine is the blue one, and my I guess we need a purple one, so we're going to do a purple one. Okay, so my cytosine is the purple. So these are my molecules here. These are my nitrogenous bases. The nitrogenous bases are just part of the nucleotides. DNA is synthesized from nucleotides, and those nucleotides, let's take a look at what those look like. Here we have my less than awesome diagram of the 4 nucleotides that are used to synthesize DNA. Let me illustrate a few parts to you. If we look here at the one, we're going to go with the one on the top left here. This structure right here that I'm going to highlight in purple is what's known as the phosphate. It is, in fact, a phosphate. You can see the P in the middle. That's the atom phosphorus, and it forms bonds with oxygen. It tends to be negatively charged, and in this case that phosphate is bound to a carbon that is part of the sugar. The sugar is ribose, except that in DNA, ribose has lost an OH group. Ribose normally looks like this. Okay, so I'm going to go ahead and draw ribose, and then I'm just going to erase that. So if I had that extra OH here, I'd be dealing with ribose. But in DNA, that extra OH is actually lost, and so we call it deoxyribose to represent the fact that I've lost an oxygen there and its accompanying hydrogen. So my deoxyribose is this sugar, which I'll go ahead and highlight in yellow so you can see this one here. And so we've got each of these corners would represent a carbon, and you can see that we have some oxygen in there. Okay. And then we've got the actual nitrogenous base itself. And so this particular one happens to be, the nitrogenous base is adenine. Okay, so this top one that I've drawn is adenine. Now, I'm not super worried that you know the exact chemical structure for this. That doesn't concern me that much. But what does concern me is, well, kind of the general shape. So adenine has two ring structures, and these double ring structures are called purines. Okay, so adenine is purine, and the chemical signal for that would be A. So we'll go ahead and put an A in here somewhere. Let's see, we'll just go ahead and spell this out. This is adenine. That doesn't look right. Let me check the spelling really quickly. Ah, I forgot an E. Squeeze that in there. All right, so this is my adenine, and it's a pyridine. Guanine is right below it, and it is also a pyridine. Okay, and again, it has that double ring structure. On the right, we have our pyrimidines, and pyrimidines have only one ring structure that you can see here. This first pyrimidine on the top right is my thymine, and then the pyrimidine on the lower right is the cytosine. Because of their chemical structures, when DNA, let's flip back here, when DNA forms its double helix and these nucleotides with their nitrogenous bases facing the center form together, we end up with the nitrogenous bases facing one another. And it just so happens that of those nitrogenous bases, we always need a pyridine and a pyrimidine to make a pair. Specifically, we need the pyridine, guanine, and the pyrimidine, cytosine, to make a pair. G and C will always form together. Likewise, we need the purine, adenine, and the pyrimidine, thymine, to make a pair. If you look at this structure, you're going to see this pattern being followed all throughout. Every time we have a G in one strand, we're going to have a C in the other strand. Every time we have an A, we're going to have a T. We call this complementary. The two DNA strands are not at all identical to each other, but they are complementary to each other. This is always followed. All right? T pairs with A, G pairs with C when we're working with DNA. Because of the complementary nature of this DNA molecule, when I separate those two strands, each strand can become a template strand for the synthesis of a brand new strand. Template, let's try that again. Template strand, for the synthesis of a brand new strand. And that's a pretty cool thing, actually. DNA is the only molecule that carries the code necessary to replicate itself. Proteins can't replicate themselves. Proteins are synthesized from messenger RNA, and messenger RNA is synthesized from DNA. So this complementary arrangement between the DNA molecules means that it can replicate itself. And that's actually one of the subjects of this lecture today. Now, I want to show you how these nucleotides pair together. And so we're going to do a little bit of chemistry here. And so what I've done is I've just copied and pasted and shrunken these down a little bit. Not perfectly, but hey, it'll do the trick. And I want to show you how if we were going to build, so let's say we have a bunch of these nucleotides and they're just kind of swimming around in the nucleus, in the fluid in the nucleus, and just kind of doing their little thing. Odds are they're not going to bind to each other. We have to have an enzyme for that. The chemical reaction would not be spontaneous at all. We're going to have an enzyme to help these guys out. And so here's what actually happens. We do have enzymes, a lot of them. And so if we were to build a nucleotide, well, let's start with a dinucleotide. We're going to start with a dinucleotide, and I'm going to move this adenine, okay, up to my top here. And now what I'm going to do is I'm going to have this guanine, and we're going to form a bond between the adenine and the guanine. Before we do this, let me give you some numbers. Okay. When we look at the sugar, we actually can look at the number of carbons, and we would have a carbon at each of these corners. I'll go ahead and just put little dots around. Each of these would be carbons. I don't necessarily write the C there, but they are in fact carbons. And for that matter, these are carbons as well. So you're aware of that fact. For the sugar, we can number those carbons. We start with the oxygen. Oxygen is not carbon, but then we move to the right. And that first carbon we're going to call the 1' carbon. This next carbon we're going to call the 2' carbon. This next carbon we're going to call the 3' carbon. This carbon that's tucked in here is the 4' carbon. And this carbon that is bound to the phosphate is my 5' carbon. So this is a 5-carbon sugar, and we can number them accordingly. We use that same numbering system for each of these. So we have my 1', my 2', my 3', my 4', and my 5'. The reason we care about this is it's going to help us understand the orientation of these sugars. Okay, so now that we've got this naming system in place, let me show you how these are going to bind. So imagine we have an enzyme, and the enzyme is going to collect one of these nucleotides out of the cytosol, the fluid within the nucleus. And it's going to arrange these so that they are close together now. So we want them nice and close together. All right. So there we have it. We now have, it looks like my carbons didn't come with it. My numbers didn't come with it. I'll just erase those and get rid of them. And I want you to see this. We have on my 3' carbon an OH. And we happen to have an oxygen here. And one of the things about these double bonds is that they sometimes are transient. So if I were to, let me draw it over here a little bit. So if I have my phosphate and my double bond, sometimes that double bond actually becomes a single bond with an extra pair of electrons. And that extra pair of electrons tends to attract a hydrogen. And generally, these double bonds will change accordingly. Because of this, do you see this? When we combine hydrogen and oxygen with another hydrogen, let me highlight that, we actually can produce water. And that's exactly what happens in this chemical reaction. The water leaves. This is a dehydration reaction. So we end up producing H2O. And these bonds change. And what happens here is we end up with, on my 3' carbon, a link between that phosphate and that carbon. We've now bound these two molecules together, and we now have a dinucleotide. Now, I'm just going to get rid of these just because they're bugging me. All right, my dinucleotide, it's a great start, but we are not happy with just a dinucleotide, so we're going to have my enzyme is going to pick up another one, and maybe we'll pick up this guy here. Maybe. Okay, we're going to duplicate it, by the way, just so I can have some more to play with. I'll go ahead and duplicate this one so I can have some more to play with. And I'll duplicate this one. Again, I just want to have some more that I can play with, so we'll just move those. Bye. Okay, so we're going to play with those later. We're going to pick one of my pyrimidines. Okay, and I'm going to move this. My enzyme's going to take this. It's going to move it into close proximity. And voila, we're going to have the same chemical reaction occur. We're going to lose water. And I'll write it the more standard way. Not that the other way I wrote it was wrong, it's just that's not the way we're used to looking at it. And so we're going to rearrange these bonds, and now we're going to have my sugar and my phosphate bound. And what do you know? This right here is my sugar phosphate backbone. That's it. That's the sugar phosphate backbone. That's this part that you can see here. That's my sugar phosphate backbone. Now, why on earth am I showing you this? Because I feel like torturing you, I guess. No, that's not it. That's never it. What I want you to understand is that when we're dealing with DNA replication, this is essentially how we're going to do it. And the reason I introduced to you the concept of 5' and 3', 5', 3', 5', 3', is to give you a directionality. It just so happens that the enzyme that synthesizes DNA, DNA polymerase, that's its name. There's actually 3 different varieties, so it's DNA polymerase 3, okay? And DNA polymerase 3 can only work by taking a nucleotide. Fish this nucleotide. Whoa, that is not what I wanted to do. Okay, try that again. We're going to take this nucleotide and move this over here. And the only way DNA polymerase works is by attaching the phosphate to the 3' end. It is not possible. Hey, I'm missing a hydroxyl group. It is not possible for DNA polymerase to work on the other side. It has to do with the shape of the enzyme and the way that enzyme works. It just doesn't happen. It's just not going to happen at all. It's not possible for that to happen. Okay. Now, so this is only one nucleic acid. It's only one. That's all we've got right now. So now I need to show you how we're going to do the other backbone. And here we're going to have a little fun. So this little thing, I don't know what this thing is hanging out there for. I don't know. Apparently I copied it. And we're going to add my OH back into this equation. I'm missing it there too. And missing it there too. Okay. Just bear with me. I'm probably going to lose this all again, so let me see if I can group these together. I'm not the greatest at this. There we go. Hey, and look, I'm even missing my line. All right, I'm not going to play with cosmetics right now because we need to move on. So you know what they're supposed to look like, and so even if some of these kind of disappear and just get funky, that's good. We're okay. So for the next strand, I am intentionally going to turn these upside down. Whoa. Now, why am I going to turn these upside down? Well, let's go up here. First and foremost, those nitrogenous bases need to interact with one another, because what they're going to do, this one is going to interact with this one, is they're going to form some hydrogen bonds. So they have to be oriented in a way that the nucleotide or nitrogenous bases can form those hydrogen bonds. My molecule does not look right. Let me look at this for a second. Yeah, they got all messed up when I copied them. That's okay. All right, so this upside down orientation, let's kind of add my little things back in there. Very interesting. Let's do another one. Which one do I want to turn upside down next? Which one didn't I turn upside down? We'll turn this one upside down. Okay. This one too. Turn it around. And that one would interact with this one up here. Okay. Again, they have to be arranged so that they can form hydrogen bonds. But of course, they also have to be able to form bonds with each other. So my DNA polymerase is going to put these together, and it is going to form a bond. Oops, I copied apparently. It's going to form a bond with this hydrogen. And so once again, we're going to have our formation of our peptide bond, or of our phosphate backbone right here, where we're going to have, let's put my phosphate back because I just deleted it. See if I can write upside down. Okay, so that was kind of a squampus bond, but you get the idea. And then if we were to put another one in there, okay, let's see, what do I need? I need a pyridine, so we're going to take this one, and I'm going to turn this one around too. Okay, whoops, go away. So around it goes, and we're going to move it in place. Okay. And now we're going to form the bond. The problem here is I actually did it backwards. My DNA polymerase can't do it the way I just did it. I took my bonds and I added to this one. I added this one, and then I added this one. That's the wrong order. If DNA polymerase was doing it, it would have to build from this one first, and then this one, and then this one. Because DNA polymerase can only add to the 3' end. Where's my 3' end? Here's my 3' end. Here's my 5' end. And my 5' end. And my 3' end. And my 5' end. And my 3' end. Okay, so let's look at the directionality of this. If we read from top to bottom, this particular strand runs in this direction from 5' to 3'. But this strand runs in this direction from 5' to 3'. We call this anti-parallel to one another. And it is simply the reality of DNA that the two complementary strands are going to run anti-parallel of one another. And then we'll get those hydrogen bonds forming in the middle. The little dotted pink lines that I kind of scribbling in there would be my hydrogen bonds. I want you to keep this in mind. We're going to revisit this idea when we talk about DNA replication. So kind of chew on it and think about it and practice it in your head, because it becomes very relevant when we're synthesizing brand new strands of DNA.